In our previous lecture, 2.1, we looked at the results that were observed by a Moravian monk, an obscure guy who taught high school biology in essence. And he made these observations, as we described before, and he showed, again, in all 7 of these traits, that if you cross these true breeding varieties with each other, only one of them comes out in the F1, but if you self-fertilize the F2s, you end up getting close to, but not exactly 3-to-1 ratio. Now, this guy made these observations, and if that's all he had done, you wouldn't know his name. You wouldn't know anything about him. No one would study him. Probably fewer people would know his name than know the name of the probably greatest geneticist in Europe at that time, a guy not by the name of Ernst Nageli. So most people don't even know that name, but you know this guy's name. His name is Gregor Mendel. And the reason you know it is because this isn't all he did. What he did is he took these observations and explained them. And that's what our goal is to do in science. We don't just observe. We explain what's happening. And so this is our first phase. You look here, we're looking at phase 1 of his study. In phase 2, what he has to do is he has to explain what's happening that's causing those patterns that he sees. Okay, now just to remind you of a few things, Mendel did nothing new. Everything he studied, every observation he made had been made before. So no one really took him all that seriously because, again, he's just a high school teacher. He's from some obscure city in Moravia, and he's not at a big university, and he's not a big research person. So what's interesting, though, is that he is the first person to suggest the correct explanation. So this is why this is so important. Now, I want to go through this carefully, and the reason I want to go through it so carefully is because it's often oversimplified, in my opinion, in the way it's taught, typically in other high schools and so forth. And we just want to be careful because there's really 2 questions he has to answer, and he does answer both, and he knows that you have to answer both of these questions. The first is, why is it that in these F2s, why are these ratios close to 3-to-1? But equally important, why are they not exactly 3-to-1? Not one of these is exactly 3-to-1. They're all close to 3-to-1. So why is that? All right, so we're going to deal with that. That's what we have to focus on here. And Mendel says this. Okay, I can explain both of those things with the following set of explanations. First, why are they close to 3-to-1? Okay, now remember, the original hypothesis that we were dealing with wasn't the only hypothesis at the time, but it was one of the main ones, was that genetics was about fluids. Fluids would mix like paints, and the paints would mix, and then you'd get something in the offspring that was somehow a mixture of the 2 parents. That usually predicted things correctly in one breeding. Okay? But he says, no. These results are telling me that's not what it is. These results are telling me that actually what these things are particles. They're not fluids that are mixing, they're particles. You put them together and they can interact with each other, but then you can also pull them apart and they're intact. And that is untrue of fluids. If you take 2 drops of fluid and you mix them together, you can't pull the 2 drops apart again. So that's his first explanation. The next thing he says is, okay, every single trait that I have studied here, these 7, now here's the point, this is critical, he's not saying this is true of every trait. He's only saying this is true of the 7 traits that he's talking about here. He was smart enough to know that, and if you read his paper at the end, you'll see he saw traits that didn't follow this pattern. Most traits do not. Most traits don't follow this pattern, which is why it is that high school students and beginning undergraduate students at American universities don't do as well with genetics as farmers do, even if the farmer doesn't have the so-called education that the student has. And it's because when we teach this, people come away with the idea that this is common and basically all traits follow it, and they don't. They're very, very rare. So he says in these 7 traits, it's particles, and for each of these traits, there's 2 particles per trait, 2 of them. One comes from one parent, and the other comes from the other parent. That's why there's 2. And one of these particles dominates. That means this. For example, let's take a look at his first study. He looked at round versus wrinkled. When he takes round-seeded organism and mixes it with a wrinkled-seeded organism, the offspring are all round. But they're a mixture of a particle from the round and a particle from the wrinkled. That's what this first, second, and third statement says. Okay, but why then is it only round? Well, because whatever the wrinkle gave it, you can't see it. It's still there. That's the key. It's still there, but you can't see it. That's what the concept of dominance was that developed after he made those statements. Okay, now basically then, these statements he was only claiming were true for the 7 traits that he studied. It turns out that one of them is still part of our modern theory of genetics. Only one. The other 3 have exceptions, but there's one that is true essentially for all traits that we've looked at, and that is this. It's not about dominance. Okay, many people think dominance is it. Okay, well, I get this all the time. People come up to me and say, well, my mom had breast cancer, and what's the probability I'm going to get breast cancer? Is breast cancer dominant? The answer is no, it's not dominant, it's not recessive, it's neither one. Dominance doesn't apply to it. It's a much more complicated trait than this. Other types of things. Hair color. True or false? Black hair is dominant to blonde hair. That's found in many textbooks, both high school level and college level. Black hair is dominant to blonde hair. That is absolutely and utterly false, and I know it's false, and you know it's false, because there's more than just black and blonde hair. If there was only black and blonde, and every time you have a mating between a black-haired individual and a blonde-haired individual, you have a high probability of having at least some individuals with black hair, then maybe I'd believe it. But the fact is there's wide ranges of hair color. So hair color is not dominant. Eye color. Brown eyes are dominant to blue. No, that is not true either. So dominance is rare. This one we have lots and lots of examples of counterexamples. What about this? Each trait is determined by precisely 2 of these particles, in every individual, one from each parent. No. Sex-linked traits do not follow those patterns. Many other traits do, but most traits don't. Human height, hair color, those things don't. Because again, those are much more complicated traits. There's only one of these statements which is still part of our modern theory, and that is this. It's particles. This is the important thing that he saw. So even though we tend to focus on this when we teach it, don't get the idea that these are common. Especially dominance. Dominance is not really all that common. These aren't really all that common either, because most of the things we call traits, like hair color, eye color, stuff like that, are determined by more than one of these particles. A bunch of particles contributes to actually what you have there. But the point is, those particles are what's contributing. It's always particulate. It's not fluid. So that's the key. This is our modern theory of genetics starts here. Particles are the determining factors of genetics. Interestingly enough, after Mendel came up with his explanation, even though we teach it today in every biology class, at the beginning anyway, when he first proposed it, it did not take hold. People did not believe it for the reasons that I discussed. It wasn't common. These trait patterns are not common, and people already had a theory that they thought they understood and believed. So in this case, Mendel writes this up, publishes it, tries to get people interested in it, gets that Ernst Nageli guy, sends copies of it to him that I was mentioning earlier, and he is nice enough to respond, but he doesn't actually believe it either, and he tries to convince Darwin that he's wrong, or Mendel that he's wrong. And so Mendel gives up. And then he goes on teaching his high school. He doesn't publish anymore, and then he dies. And then 35 years later, at the turn of the 20th century, 3 different biologists doing different types of experiments actually rediscover Mendel's work. And they say, yeah, he's right. And Mendel never knew it. So Mendel never knew how important his work actually was. Now, since that time, however, we have done lots and lots of experiments to confirm what he did, but also we've added new concepts and new terms. I want to introduce those terms now so that even though we're talking about what Mendel is doing, in fact, I'm going to present Mendel's results in his full paper here shortly, I want you to use the modern terms and think about it in terms of the modern terms. So here's just a few things that we need to understand. What are the particles that Mendel was talking about? Well, the particles are genes. That's what we call them today, genes. Now, the concept of a gene is kind of fuzzy at the moment. We used to think we understood it. Concept, yeah, the particle is the gene. And your book says a gene is the basic physical and functional unit of inheritance. It's an ordered sequence of nucleotides located at a particular locus that specifies a functional product. Turns out that that isn't strictly understood to be true anymore. The gene is definitely a basic physical and functional unit of inheritance. It still acts like a particle. However, it isn't necessarily in one location on a chromosome. there are these regions, which we're going to study later, called enhancers, which we know now from fairly recent research tend to be scattered all over the chromosomes, and they all impact the function of a particular gene. So we tend to think of them as regulator elements. We're trying to keep them separate from the gene itself, but then the gene also has other regulator elements called promoter, and so which is important in terms of the gene and which one is a separate regulator of the gene, we're arguing about. Doesn't matter. The fact of the matter is this still gene concept is still a functional, important concept because it is still the unit of inheritance. That's what it is. It's the particle. So when we talk about a locus, though, we say that it's in a particular locus on a chromosome. We say it's in a particular spot on the chromosome, and that's what a genetic locus is. It's just a particular spot on a chromosome. So here's an example. This is human chromosome number 7, and there's a gene that I want to know where exactly it is. That gene, for example, that we're using here is called CFTR. It's a gene that's important in cystic fibrosis. It's called a cystic fibrosis transmembrane regulator protein. And this particular protein is found in this exact cytogenetic locus right here. Not the protein, but the gene for it. Now, when I'm talking about the gene here, what I'm talking about is none of the regulatory elements. I'm only talking about what's called the structural portion of the gene. We'll discuss that later. But it's found here, and this is how we designate its location on a chromosome, 7q31.2. Now what this means is this. If you look at chromosome number 7, it has a centromere, like I've described in lecture 1.6. Now every chromosome's centromere is slightly off-center. No human chromosome has a centromere dead center right in the middle of it. There's always one arm that's slightly shorter or quite a bit shorter than the other. The short arm we always refer to as the P arm. Now this is going to be important in the laboratory, the mitosis laboratory that's coming up. So make sure you see this before you do that lab. But there's the P arm right there, and then the Q arm. The Q arm is the longer arm. Okay, so chromosome 7 has a P arm short and a Q arm long, which is the arm is simply the distance from the centromere all the way out to the telomere. All right, when we look at chromosomes, we often take dyes and band them with a particular type of dye, and that's what's been done here so that we can sort of recognize exactly what section of the chromosome we're talking about. Now, when we talk about the location of this gene, the gene for CFTR is right here on this chromosome. There are a bunch of other genes on here, literally thousands of other genes on this chromosome, and we're going to study that sort of organization here shortly. But the point is right there is where this CFTR gene is, and it's on the Q arm of chromosome number 7, so that's why we say it's on 7q. It's on the long arm of chromosome 7. But the long arm of chromosome 7 is broken up into pieces. For example, right here, the last part of this arm is a region that we call number 3. So that's chromosome region 3. There is a subband right in here that we call 2, which is a subsection of region 3. And there is a band that's bigger. Sorry, the subband is this little one. The band is bigger here. We call that number 1. So this designation, 7q31.2, literally means chromosome number 7 on the Q arm, long arm, it's in region 3, band number 1, subband number 2. That's how we read this sort of a system here. So this is the way we now understand Mendel's particles. They're parts of chromosomes or pieces of chromosomes on which particular structural information about how to make a protein is found. Now, that's not the only thing genes do. Genes do a lot of other things, or chromosomes, and DNA does a lot more than just to determine the structural proteins and the structures of proteins. It also determines how much of the protein is there. We'll get into that later on, but for right now, I want you to be thinking of these as Mendel's particles. Okay, with that terminology, we can now restate Mendel's statements, Mendel's explanation for his observations, using a little bit more modern terminology. So here they are. What Mendel claimed for the traits he studied, just the 7, each one was determined by genes. Those are the particles. And that's the part that is still part of our modern theory of genetics. And this is the only part that's really ubiquitous in our modern theory of genetics. But in those 7 traits, he also says that each trait is determined by 2 copies of the same gene. And 1 copy comes from one parent and 1 copy comes from the other parent. Then there's this other concept that we have to have. And remember we said that one of these forms of the gene dominates the other. So the form of the gene for seed shape that makes a round seed dominates the form of the gene for seed shape that makes wrinkled seeds. So in this case then, one of these gene forms has to have a name. We call the different forms alleles. Okay, and so the definition of an allele is an alternative form of a gene. Now this is what's important. A gene is something that we all have. All plants, for example, all pea plants have a gene for seed shape. They differ in their alleles. So this is kind of critical. We often say things differently, and this is perfectly fine. You can say it this way if you want, but technically we don't mean it this way. You can say things that you and I are different because we have different genes. That technically that's not correct. You and I are not different because we have different genes. In fact, you and I are human, therefore we have the exact same genes. We have the same genes. We have eyes. Those eyes have colors. We have the genes that make those colors. The difference between you and me is not that we have different genes. The difference between us is because we have different alleles of those genes. So the alleles are the various forms. And again, one form in Mendel's example makes round seeds, another form makes wrinkled seeds, but it's only one gene. The gene is for seed shape, and there are these two different forms for it. So we have to keep that in mind because that's different than the way most people talk, because it's something that they don't really run into. They don't really think about it in terms of those details that we have to think about as professionals. Okay, so that's the thing. One of the alleles is dominant and the other is recessive in the traits that he's talking about. Don't forget, I'm going to keep hammering this. This concept of dominance and the other one being recessive is something that is not common in most traits. Okay, so that then is a statement now as we would say it in modern terms. But there's a bunch of other terms that we have to have in order to make our lives a little bit easier in talking about these things. These are terms that you've probably seen before, maybe not, I don't know. But I want to make sure we have the technical definitions down. First of all, when we talk about an individual having two particles for a trait, and if those two particles are the same type, then we say the individual is homozygous for that trait. What we mean, for example, is this. Suppose we have a pea plant and it's got round seeds. Now remember, that round seed is made by two different genes. They're the same type of gene, but one came from the mother, one came from the father. And if they both of them make round, if both of those are the round alleles, then we say the individual is homozygous. If, however, that round individual has a round allele and a wrinkled allele, its seeds will be round because round is dominant to wrinkled. And then you'll have what we call heterozygous. You have two different forms of alleles for a single trait. So we can be homozygous or heterozygous. Here they have both the same forms. Here they have different forms. And then there's this other concept, genotype, phenotype. When we talk about these forms, I'm going to make this easier to understand here when we use notation here shortly. But when we talk about the forms of these genes, we talk about this allele being the round allele and this allele being the wrinkled allele, we will be talking about their genotypes. So we're talking about the list of alleles that an organism has for a given trait. That's the genotype for that trait. How the trait actually looks is the phenotype. So that's the observable property. Okay, so for example, if we talk about a seed being yellow or hair being brown or a person being tall, that's the phenotype. Right? So those are the different concepts that we're going to be using here as we move ahead and start looking at these things. But there's one other concept that we have to look at, and that is this concept of dominance. Dominance seems to not mean what most people think it means. It means something very, very precise. And I want to get into that. I'm going to show you that as an example. So to help me explain what dominance actually means, I'm going to enlist some famous people. Here are some of what I consider some of the most famous people on the planet. Actually, this gentleman is no longer around. He's no longer alive. But this is Neil Armstrong. He's the first person ever to set foot on the moon, and this is his partner Buzz Aldrin. He is the second person to set foot on the moon, and this is Michael Collins. And he is the person who orbited the moon while these guys were walking on it. This is the crew of Apollo 11. And if you look carefully at their chins, I want you to focus on their chins. And so you look here, there's some variation how they look, but those chins are much like mine. I call those normal chins, but that's not trying to denigrate anybody. I'm just calling it normal because that's what I have. I want you to compare it to this guy. I want you to compare their chins to this guy. You might recognize him as Aragorn from the Lord of the Rings trilogy, and this is Viggo Mortensen. And you see that he's got a nice dimple right there. We call that a mid-chin fissure or a cleft chin. And the question I'm asking is, which of these is dominant? Is the cleft chin dominant, or is the non-cleft chin dominant? Turns out that actually makes sense. It turns out that this is one of the few traits in human beings that actually follows Mendel's principles quite strictly. So that's our question. Which one is dominant here? Well, if you think about it, which one is more common? Is it more common to have chins like this, or is it more common to have a chin like Viggo's? That's the question that I want to answer before we decide whether or not one of these is dominant or recessive. Okay, so if you look around and you think about it, you'll find that the cleft chin is much more rare than a non-cleft chin. So most people would say, well, the cleft chin is recessive because it's rare compared to the other. But actually, if we look at the definition of dominance, we'll find something interesting. Any allele that's expressed in a heterozygote is dominant. Meaning this, suppose you have an allele for a gene for chin shape that makes a cleft chin, and you have an allele for chin shape that makes a non-cleft chin. Whichever one of those is expressed in the heterozygote is going to be the dominant one. Okay, so by just looking at which one's more common, we would say non-cleft chin is dominant because it's more common. But we would be wrong. It turns out that the cleft chin is the dominant one. Whether or not something is rare or common has nothing to do with whether or not it's dominant or recessive. It's simply this definition. That's the only thing that matters. So how do I know that the cleft chin is dominant? Well, if you have an allele that makes a cleft chin and another allele that makes a non-cleft chin, together you end up with a cleft chin. And therefore, by this definition, the cleft chin is dominant. Okay? So any trait that is not dominant, we call recessive. So so the trait or the allele, if it isn't expressed in the heterozygote, technically it is not dominant, it's therefore recessive. Okay? Now here's an example. Let's go back to garden peas. And we're looking at the round and wrinkled phenotypes. And we know then that each of those phenotypes is determined by two particles, one from each parent, according to Mendel's explanation. And we're going to say there are two alleles that exist. And we're going to call one of them big R and the other one little r. The big R is going to be the allele that makes a round seed. The little r is going to be the allele that makes a wrinkled seed. Now, I want to point something out here. We are not going to say big R and W. The reason we're not going to do that, even though some textbooks do, is that goes against convention. Our convention is that the gene has a particular symbol, in this case R. The alleles have variant forms of that symbol, big and little. Okay? Now when we talk about real genes, we generally don't talk about big R, big A, little a, or whatever. We have different symbols, and I'll introduce those later in the course. But for right now, the gene itself is R, and it has two alleles, big and little. The big one makes round, the little one makes wrinkled. And therefore we can have three possible genotypes. Remember, the genotype is how they're combined. So I'm going to get two of these for each trait. So it could be big R, or it could be big R, it could be little r. Now big R, both are carrying round alleles. Therefore the seed shape for that individual is going to be round. Little r, same reasoning. Therefore the seed shape is going to be wrinkled. But what about this one, the heterozygote? Big R, that's the one that has two different alleles, so it's heterozygous. What about that? Well, by dominance, we know from Mendel's experiments, in his hypothesis, the round would have to be dominant, because when you take a purebred round and a purebred wrinkled, and you breed them together, you must have a big R from one parent, a little r from another parent, and therefore the individual in the F1 must have each, big R, little r, must be heterozygous, but they're always round. Therefore, round must be the dominant trait, which means that this individual must have a round phenotype. Therefore, round is the dominant trait, and therefore also big R is the dominant allele. Okay, so let's practice a little bit. I want you to answer these questions on your own. Don't go ahead, don't look ahead at the slides, don't look ahead at the videos. I want you to answer these on a piece of paper separately before you see the solutions. Okay, start with this. Plant has wrinkled seeds. We're talking about a pea plant. We're talking about Mendel's experiments here. One of Mendel's pea plants has wrinkled seeds. What's its phenotype? There's another one. A plant has wrinkled seeds. What's its genotype? This will give you practice in differentiating those two concepts. A heterozygote has what phenotype and genotype? If a plant has yellow cotyledons, now cotyledons is the seed itself, the parts of the seed, the two leaves that make up the seed. If they have yellow cotyledons, what's the genotype? Okay, answer those questions, and then we'll move on to the next segment.